we tend, we tend to focus on helping people with advanced disease, which, over time, I have realized is probably not where we should be focusing our efforts, right? If you have advanced disease, that is something that's very hard to turn around. We might be able to help you manage it, but it's going to be very difficult to cure you. And so I've kind of arrived at this perspective. It's also turns out to be a central mandate of one of the organizations that I work with, the Zuckerberg Initiative. But I've just really arrived at this conclusion that we might be a lot better off thinking more about disease prevention. And so I'm sure you're all familiar with this quote. A very wise man, Benjamin Franklin, said that, if we had an ounce of prevention, it's worth a pound of cure. I never knew anything about the context in which he actually said this. He was talking about protecting towns from fire. So he was saying, like, it's a lot easier to prevent a fire than deal with the aftermath of your town burning down to the ground. But we think about this now, right, in the context of disease. And I think this is really an idea worth thinking more about, just in terms of how do we use technology to prevent advanced disease? How do we, get in there at an early stage where you could deliver something that would be more like a cure or just complete prevention of something happening to a patient. So I'm going to give you some stats that really reinforce my belief in this, and I'll ask for your forgiveness. So these stats are from a report that was put out by McKinsey. I want you to rest assured I did not get all of my scientific information from McKinsey, and I have read the primary literature that this was based on, and I think these stats are pretty solid. But this is a pretty staggering one, okay? So we know a lot about preventing disease. We don't use a lot of the things that we actually know how to do. And so if we used known interventions, things that we have at our disposal right now, we could reduce the global disease burden by over 40%. And what that means, this is all kinds of things. It's an infectious disease, right? There are parts of the world where infectious disease is not under control. We lose people to curable infections. But there's plenty of developed world problems also squarely in this bucket. And if we did a better job, if we used the measures that we have at our disposal, 230 million more people would be alive right now. And we all have people, in our lives, in our families, in our networks, right, that are part of this loss of life, right? People that get diagnosed with disease too late. Cancer is a classic example. You're stage 4. It's really hard to treat and deal with. And so this statistic just really, it blows my mind. 230 million people that would not have died from basically preventable medical issues. And really 70% of this is prevention, environmental, behavioral, social interventions, preventive health measures. Okay, so there's some other interesting statistics here. Okay, so if we did prevention better, 65 would be the new 55, right? So we kind of reaped 10 years. I personally decided to stop aging at about 38. I don't know about you guys, but that was just kind of the time that I thought, you know what, I'm done. So I'm just parking myself squarely there. But if that doesn't work out for me, then I really like this, right? To be 10 years kind of ahead of the game sounds awfully good. And this is, the thing that maybe blows me away the most, that there is a huge impact on GDP that comes from preventing disease, that comes from those 230 million more people that are on the earth. So, why are these things connected? Okay, we have fewer early deaths, fewer health conditions, expanded participation in the workforce. We lose people out of the workforce because of health issues. And these are actually our people that are very productive, huge amounts of expertise, super skilled, and we kind of lose them almost when they're the most useful because we're just not that good. You know, people have vision problems, hearing problems, back problems, all kinds of things, and they exit the workforce early. And so all of this, and even taking people and increasing labor productivity, that benefits the global economy by $12 trillion. So, you know, that's just a staggering amount of resource, right, going into the global economy. So this is a health story, it's a societal impact, but it's also just an economic story as well. And if we did prevention better, it means that for every dollar that's invested in better health, we get $2 to $4 back because of that, you know, reinforcement of the workforce and all of the other good things that happen. Right now, we put $1 into healthcare, and it just, you know, it evaporates. Our healthcare system does not care for our health right now, right? We use it to care for ourselves when we are sick. So that these are all parts of you know, one big problem. Okay, so you may be asking yourself, why is she talking about this? What does this have to do with science? Well, it has everything to do with science, because as scientists, we have this incredible opportunity to be the ones to develop new technologies. I mean, maybe some of the things that we could use, we don't use because they're too hard to use. They're hard to kind of get into the mainstream. People don't like them, or they're unpleasant. So here's a great example of something that has everything to do with science. And after I get through this, I am going to take a turn squarely into our science. And so this is where technology is really starting to make a dent in the management of a chronic disease, that's diabetes. Okay, so continuous glucose monitoring. So glucose monitoring, right, which is absolutely critical to managing diabetes. It used to be, I don't know how far back this goes, maybe in the 50s, you had to go to a pharmacy or a doctor's office to get your blood glucose measured. You didn't do that very often, and then you just kind of guessed the rest of the time. Like, oh, I kind of feel okay. I think I'll just keep taking my insulin, you know, a lot of guesswork. Then we got the handhelds, right? Some of the first really practical sensors that were developed were glucose sensors. And after decades of work and engineering and development on those glucose sensors, then we had these devices that people could use anytime, anywhere, prick their finger, measure their glucose. And all of a sudden, the management of diabetes got a lot better. You know, guesswork was gone if you chose to use one. Okay, so then turns out, and I'll get more into this, that the technology that is used for glucose sensing is something that you can put on a little microneedle, attach it to a patch with a bunch of inexpensive electronics, and you can put that little needle right into your interstitial fluid, let it sit there for 2 weeks, and continuously measure your glucose. So this is another big leap forward in technology, and this is kind of how it connects back to maybe not prevention, but certainly better management of disease. And so the use of these continuous glucose monitors is projected to save the healthcare system billions, help diabetics very significantly, because when you're diabetic, right, if your glucose levels aren't totally under control, that does a bunch of other bad things in your body. You get inflammation, there's vascular issues. You know, people that have uncontrolled diabetes, they can lose a limb, right? That's because of the blood flow. And all of these things, all the comorbidities, the kidney failure, all of the other things that go hand in hand with diabetes, those are all projected to kind of be much more manageable with this tool. And then even the other step up, right, is integrating an insulin pump right there with a continuous glucose monitor, right? So you're measuring, you're administering, and then the disease is really under control. And then you don't have all these other issues. So I think this is just a great example of how if we really push technology, right, we can conquer diseases that have been really debilitating in the past. Okay. So that's the kind of long-winded introduction. So now let's talk about this glucose sensor. Again, it's a patch that has the electronics that little needle goes right into the interstitial fluid. If you've never heard of interstitial fluid, it's, if you, for those with kids, or maybe you've done this to yourself, you skin your knee, you don't quite get to the blood, but it's like there's water coming out of your skin. That's your interstitial fluid. It's sitting right there. It's in equilibrium with the blood. And it's a really nice place to put a sensor because you don't risk infection at nearly the same level as if you were to go into the blood. So sitting in the interstitial fluid is a nice place to be. And this glucose sensor, so basically the way that it works is you take an enzyme called glucose oxidase that gets mobilized on this microneedle, which is basically an electrode. And the glucose oxidase it's a naturally occurring enzyme. When glucose reacts with it, there's an electron that trades hands. And so having that enzyme on an electrode means that you can tie an electrochemical current with the concentration of glucose. So that's a nice, easy way to do that type of quantitation. So this is something obviously super robust, has been miniaturized to a point where it's wearable in the arm. Okay, so why don't we have continuous glucose monitoring for like everything, right? Like there's so many other biomarkers that would be incredibly helpful and maybe a step towards preventing disease if we could do this. Well, it turns out we don't have glucose oxidases for cytokines or DNA or other molecules that would be of interest to track. And so that's the big unmet need. And that's what I'm going to tell you about today is kind of how we're starting to address that unmet need. And if we can find a way to kind of come forward with other continuous monitoring devices, then we don't just have this capability for diabetes. We could use it on some of our advanced chronic diseases that are huge burdens to our society and our healthcare system, including cardiovascular disease, especially heart failure, which is one of these things where it's really hard to tell what's going on with a patient on a given day, very hard to manage, a lot of hospitalizations for heart failure patients. Kidney disease is same thing, difficult to manage remotely. And then the area that the Biohub focuses on that I think is really key to all of this is inflammation. And inflammation is really, that's the thing in many cases that gets disease started, right? Your immune cells are kind of seeing something that they don't like, or they think they're seeing something that they don't like, and then they start to actually damage healthy tissue. And if we could better, spot that, and turn it around, there are many, many diseases, different kinds of cardiovascular disease, some of our cancers, neurodegenerative disease, that we could really take the burden way, way down. So that's an area where we really want to get a handle on, physiology at the molecular level. Okay, so now we'll kind of get into the hardcore sensing side of it. So this just kind of gives you a sense of where the field has been, where it's going. Back here in the 60s, this is when the glucose oxidase stuff started. Oxygen was another popular sensor back in the 50s because it's just direct electrochemistry. And then there has been, an explosion of different things that have happened in the late 90s. This is what I worked on as a PhD student at Caltech. Not to date myself, but that's, if you look at the introduction of my thesis, I think that figure is in there, where we tried to make electrochemical sensors for DNA molecules and did some good work there. And this field, so when I left Caltech, I did not think that I was ever going to work on an electrochemical sensor ever again, because I really didn't want to. For the students in the audience, you know how you feel about your PhD work by the time you're done. You're like, I'm out. I want to go do something else. And this is a long story that I won't tell right now, but like electrochemical sensing, it's like the younger sister that follows you everywhere for me. Like it's something I started my career, I wasn't going to work on it, and before I knew it, it was just following me, and I could not get away from the field. So here I am, many, many years later, but glad that to have stayed in it. And actually, one thing that happened kind of in here, the field was actually starting to die. because all of this work was going on. The glucose stuff was getting really exciting. None of the other stuff was getting anywhere. Like we thought we were going to make all these clinical diagnostics based on electrochemical sensors. There were protein sensors, there were DNA sensors, but it was just really hard to kind of break through. And then what happened is that the chemists like me, who knew how to make electrochemical sensors, and then the engineers kind of got interested in this field. And they started thinking about making devices. Like, how do we take sensors and start putting them into really interesting new form factors to potentially enable this kind of continuous sensing? And it really injected new life into this field. And now it's kind of exploding, which is awesome. So if you're in a field that you feel like is getting a little quiet, hang in there, and these things can happen. And so there's a lot of really interesting devices that are out there now, but they mainly detect glucose. That's the one thing. So the engineers, without a new sensor, right, they can't really do much except create all these really interesting form factors, little glucose sensors you can put on your tooth or in your contact lens, but not necessarily new chemistry. Okay, so that's what my group has been working on for decades now. Is developing new types of sensors to bring new capabilities into the field. We have done a lot of work on nucleic acids. We've done a lot of materials chemistry as well. One of the things that's really tricky with these sensors is just getting enough sensitivity to actually see something in a clinical specimen or in a human specimen. So one of the things we spent a lot of time on is creating materials that kind of allow us to bring the sensor to the molecules, because one of the things that limits sensitivity is that molecules diffuse really slowly, relatively. And so, if you want a molecule to diffuse to your sensor, it takes a while, but if you bring the sensor out to the molecule, it takes less time, and you can gather up more molecules if they're quite dilute. And so that's a lot of what we've done. We've used these reporter systems that are what we call electrocatalytic. That just means you have a couple of ions. One of them does a reaction that is electrochemical, so you're sending electrons around electrochemically. The other compound kind of absorbs the electrons so that you can make everything catalytic, hopefully produce more sensitivity that way. And this is a picture of one of our, we call these nanostructured microelectrodes. It's a nice spiky electrode made out of gold. And these systems, so I'm going to take you through just a few minutes of the early work, just so you can see kind of some of the limitations of conventional types of assays. So these types of sensors where you have electrochemical reporter groups, the way that you use these is maybe you have a piece of DNA, you're looking for another piece of DNA, it's going to bind, you're going to put your redox reporters in, you're probably going to do a lot of washing in between all the steps. This is not something you're ever going to put on a microneedle and put in somebody's arm, right? Because you can't squirt the reagents into the human. So it's a little bit of a dead end, but it certainly inspired us to kind of go and do some other things. These types of systems, though, we have done a lot with them. So in more of a format that you would think of as like a clinical diagnostic type device, we've done things like develop assays for blood-borne virus detection or liquid biopsy, looking for nucleic acids in the blood that come from tumors. We've built assays for small molecule drugs of abuse or antibiotic-resistant bacteria. This was one of my favorite projects. We teamed up with a team of lung transplant surgeons that needed a way to quickly assess lung tissue. So when a lung comes out of a donor, the surgeons have to figure out whether to put it into the recipient. And I think it's about 60% of the time they reject the lung because they're worried. If you put the wrong lung into a recipient, you're going to cause more problems than you're solving. And so we built them a device that they could use to look at molecular markers to figure out if the lung was healthy enough to be transplanted. So we did a lot of good things, even with our early systems that were a little, kludgy or whatever. And we also spun a company out. Zan noted our patents, and that's kind of our typical workflow. We create technologies, we make sure they're really robust, we test them out on a lot of different applications, we patent, and then if everything looks good, we try to either partner with a larger company or start a new company. So this is one that spun out of our group, and the company went, from our very early research grade technology all the way to a commercial product. And they built a testing cartridge around the electrochemical sensor technology, built a little benchtop analyzer. It was all sample to answer. You could put a swab into the cartridge and get a yes, no. This was an infectious disease product. Cartridge was less than $10. The instrument was $1,000. 20-minute turnaround time. This was all going on in like it was early 2010s and kind of into 2015, 16. So if you think about it, like very much pre-COVID, like this looks like the solution that we eventually got, right, at the pharmacies for COVID. We were a little ahead of our time here. It was actually very hard to convince clinicians that they wanted to do their own testing in their offices. So we ended up selling the company to General Atomics, which is a defense subcontractor, and so they took it out kind of through military channels. But the thing that I learned from developing or being involved in the development of these consumables is that these reagents, even for clinical diagnostics, it's a big pain. Like all the little pellets of the redox reporters and everything, really complicated to scale and make robust enough for commercialization. So that, coupled with this desire eventually to develop implantable devices, had us kind of take a turn. And that's when we started working on sensor technology that we thought might be amenable to eventually developing implantable sensors. So, if you look around, so this is kind of back to the kind of engineering-driven devices that have been emerging over the last few years. Again, mainly glucose, things like lactate, a few other things. And again, it's because of the lack of sensor technology that allows you to reagentlessly, this is really critical, you got to get rid of the reagents to do this, has been a big limiting factor. So there have been implanted electrochemical sensors that basically do electrochemistry on small molecules directly. So you can take like a carbon fiber, put it in the brain of a rat. If you know the signature of a particular neurotransmitter, you can kind of watch it. It's hard. You have to have something that's redox active. And then the other main class of sensors were developed by Kevin Plasko and his group at UCSB. These are little aptamers that when you bind a small molecule, they change conformation, and you can detect that without any external reagents. They really only work well for small molecules, though, so they don't give you the coverage of the protein markers that we really wanted to get at. Okay, so we came into this effort, and one of the first things we did, so one of the things I love about working with engineers is that they're obsessed with requirements. Like, what are we actually trying to do? What does this thing have to have for us to be successful? Chemists don't work that way. We don't do that. We just run into the lab, we try a bunch of things, and then when something works, we kind of back out, whether it's going to be able to take us where we want to go. The engineers don't do that, right? They say, okay, we got to make a sensor that is reagentless, no added components. It's got to be compatible with biological fluids. We want to put this into a person someday. It has to be amenable to continuous monitoring. It's going to have to be able to regenerate itself. And we really want it to be adaptable to any analyte. We're going to go through all this trouble. We want to be able to make a sensor for one protein, make a sensor for another protein, and put it all together. So that was what we kind of started out with in terms of what we needed. And what I'm going to take you through now is kind of like we worked on this problem for, I would say, almost 5 straight years with very little in the way of success. It's a hard problem. Like trying to have everything attached to each other, nothing coming in from the outside world, you can't wash the sensor, it's a lot of hard things to do. So again, for the trainees in the room, like don't forget, like when you hear people like me talk, we're only telling you about the stuff that worked. We never tell you about the stuff that didn't work. And you're in the lab probably mainly generating negative results. That's how science is. But here's the stuff that finally did bear fruit. And so this is a class of sensors that were developed by Geronimoy and Strathmite group. And they came up with this name. They refer to these as inverted molecular pendulums. I have learned to not like this name for these sensors because if I get in front of chemists and there's a few physicists in the crowd, boy, do I get a lot of grief about calling something upside down a pendulum. It has no momentum. It has no, So anyway, I know you guys won't hurt me over this, but all I want you to think is like an arm, a bob, and it is going to swing, not with momentum, but it is going to swing. And so these pendulums, there's a lot of interesting things going on with them physically, and I'm going to tell you exactly what they are in a minute. But if you think about just the construct, right, we're going to attach these to an electrode surface. There's going to be a drag force, right? If these things are moving around on the electrode, there's going to be drag. And let's say we had a way to increase the drag on the bob of the pendulum, maybe something bound to the bob. Well, we should be able to see that because drag slows molecules down, right? There are coulombic forces between the pendulums because they are charged. But then the other key thing is that we're going to put an electrical field on the electrode, and so there's going to be an electrical force, okay? So that was how we thought about putting these pendulums together. And we built a really nice model and then started to simulate electrochemical experiments. So this is a chronoamperometry experiment where you pulse a potential and you measure the current transient. And we could see in our simulations that for the unbound sensor, so where the bob doesn't have anything attached to it, this is what the transient looks like. It kind of recovers pretty quickly. For the bound pendulum, where we've increased the size of the bob because something's attached to it, we see the kinetics slowing down because the motion has been slowed down. So this looked pretty promising computationally. And this is like just a very simple paper and pencil model. And so now when we built the pendulums, this is how they're put together. So we have a piece of DNA as a linker, small piece of DNA, so nice and rigid. So that's the arm, also negatively charged. So if we're going to put a field, let's say on the electrode, and we're going to try to attract the pendulum so that we can kind of synchronize the motion of the pendulum, we can do that because we've got our negative charge here. We can put a positive field here. We've got an antibody attached to the DNA linker. That antibody is going to specifically bind whatever it is we're interested in. And then we also have this ferrocene molecule, and that is what we're going to use to detect the motion. So ferrocene, once it gets close enough to the surface, we can oxidize it. So we can pull an electron out of it. And so then what we're looking for is the difference in the movement of this form of the sensor versus a form where the protein has bound to the antibody. And you can see it's reagentless, right? Everything's built in. Maybe the one thing that traditionally would have been a reagent, the ferrocene, we've actually attached it to the sensor so you can just anchor everything before you get going. And, lo and behold, when you do the electrochemical experiments, you can see that this works as predicted. It's a little bit different. The curves are a bit different. In electrochemical experiments, you have Faradaic current, which is electrons moving back and forth. You also have capacitive current, which is just ions moving around. And so you get this kind of biphasic profile, but you can still see a very healthy difference between bound and unbound. And, we just put the sensor through its paces just to make sure that it would meet those other requirements. The sensitivity of these systems is directly related to the antibody that we use to bind the analyte. If we use an antibody that has really high affinity, we get high sensitivity. If it has low affinity, we get low sensitivity. Here we're looking at cardiac troponin. We have good antibodies, so we can get down as a picogram per mil range. We looked at different biological fluids, and this is one of the things that's really tricky in sensor development. Very often when you go into something that's like really complex, has a lot of biological activity, things fall apart. But I think here, because we're moving the sensors around, right, we apply the potential, the pendulums start to move. I think that really helps us with the complexity of some of the, potential for fouling in there. And then we also did a panel where we developed different sensors. We swapped out the antibody, put another one on with different specificity, and showed that we could look at a variety of analytes. We were kind of ticking off those requirements as we went. And we also showed that these were good sensors for continuous monitoring. So here we have one of our sensors. We're just sticking it in a beaker, just like a pH meter, and then bringing in some troponin. And you can see that we inject it, and then the current modulation kind of kicks in, and then it plateaus. So we've got what we need to do continuous measurements.
I have a question.
Yeah.
You're taking an antibody, which a full antibody is 150,000 molecular weight.
Yeah.
And you're adding troponin, well, that's reasonably large, what, 15,000?
Yeah.
And that's going to make a difference in the overall,
It does. It's small. The smaller the analyte, the smaller the change. We have over time kind of like tried to use the most minimalist antibodies that we can find, Fabs, that kind of thing, so that we're not dealing with a huge, so that's part of it. But if you look at this paper, we actually did the simulations as a function of different deltas in terms of the hydrodynamic diameter of the complexes we were making. And yeah, there's a little bit of modulation that you can get from a 10% change or a 20% change. Yeah. Okay, so then to cap this first set of work off, we did an experiment where we tried to do continuous monitoring in the mouth of a mouse. This was like just a typical PI level idea. Like I remember bringing, Sarath and Gadamoy, the folks working on this project in my office, and I said, guys, for this paper, like to get into a really good journal, we have to show that we can do something like in an animal. And I said, we gotta like really hurry up and get this paper out. So I don't want to do anything too crazy. Let's just like make an electrode array. Let's put it in the mouth of a mouse. We'll inject the analyte in through the tail vein, and we'll watch the analyte come into the saliva. And I could see the looks on their faces. They were like, this is not a very good idea. And I said, it's going to be fine. Just go try it. Let's just see if we can squeak out some data to move forward with. And I could see they like left my office. They walked down the hall, and I could see they were huddling there. And then they came back, and I could tell they were really like on the fence about whether they should broach this with me. But they said, Shana, have you looked at the mouth of a mouse recently? And I said, no, I don't think I ever have. And they said, well, it's really small, like really, really small. So we got to make something that's really, really small. I said, guys, I know you can do it. Get in the clean room, make a little electrode array and make it happen. And there was a lot of eye rolling and, unhappy graduate students, but they did it. They made these really little arrays that were just small enough that we could kind of tuck them into the oral cavity. We did the tail vein injection of troponin, and we could see it coming into the saliva. So that was a win just in terms of being able to do this kind of measurement in real time. Okay, so that was what got us going. We've done a lot of other things with these sensors. So back to this drag question that was just asked, like, here's a really nice one, right? Like no problem having one of our little pendulum sensors grab a viral particle, right? There you can see there's a ton of modulation. So we did some work on SARS-CoV-2. We've done things like combine with the coffee ring effect to help with sensitivity and concentration. We've also tried to get rid of antibodies when we can and use aptamers instead, so that then that bulk problem really goes away. And then we do have a startup that's making these little handheld devices, kind of like the handheld glucometers, but for protein biomarkers. But so that was all the foundational work. And so now it was really time to like get serious about continuous monitoring here and not do the easy experiment like measuring something in the mouth, but instead, doing what has worked in mainstream clinical medicine and, really almost for consumer grade products, and to bring a sensor into the interstitial fluid to do continuous monitoring. But there was another really big problem that we had to solve. So you know, in our mouse mouth experiment, right? You may have noticed we went up in concentration. We did not try to come down. And the reason we didn't try to come down is because we actually knew that we couldn't. And that kind of caused us to turn into a different problem and has led us to some really interesting science and a sensor that is now really fit for purpose for continuous monitoring. So the problem here, right, if you want to do continuous monitoring, right, like the glucometer, it measures glucose levels going up, it measures them coming back down and up and down and up and down, right? You have to be able to do that to make a useful measurement. But glucose oxidase and glucose, right, that's an enzymatic reaction. The enzyme knows to let go of the glucose once it's been oxidized. Not the case in our sensors, right? Our sensors are affinity based. And we use really high affinity reagents so that, you know, when a protein is floating by, the sensor grabs it, right? But the problem is it doesn't let it go, right? So concentration can be, you know, going up, the sensor's got an analyte, concentration comes down, well, equilibrium, you know, is going to take a long time just given the affinity of that complex. So if we're using a receptor, let's say it has a KD around 1 nanomolar, we're usually down in with picomolar KD values. And so for a picomolar KD, right, what does that mean? Like really high K on, very slow K off. And so K off means it's going to take about 20 hours for a sensor to respond to a concentration going down. So that really takes this whole thing and makes it not that useful if we want to detect real-time changes in physiology. So this problem, we kind of recognized it. I think the field had also recognized it, and there just wasn't a solution out there because you could make your antibody, you know, you could kind of take the KD down, but then you're going to lose your sensitivity as well. So that's kind of a non-starter. Okay, so the guy who really saved the day here is Hussein, who's just a really talented postdoc in my group. And he really, you know, he got deep into this problem. Here's a set of data that shows you exactly what I'm talking about. So we've got a sensor. This is for myeloperoxidase, which is another cardiac marker. He injects 200 picograms per mil. The sensor plateaus. That black arrow, he took the protein away and just replaced the protein solution with buffer. And you can see what happens, like nothing, basically. The sensor's sitting there with the protein bound and it's not letting go. You know, if you use a sensor with a lower affinity, right, you can do the same thing and you start to see the signal come down. It kind of plateaus, which always bothers us. There's a variety of things happening there. But that sensor, if your KD is 27 nanomolar, it doesn't really tell you anything clinically relevant. And so Hussein came up with the idea, and this is really the essence of the science paper that came out today. He said, Shana, what if we take one of these sensors where we have a protein bound to the sensors, and what if we shake it? We shake it hard enough, maybe the analyte will come off. And I said, okay, Hussein, how are you going to shake the sensor? He said, well, we're on an electrode. I'm going to oscillate the electric field, and I'm just going to wave it up and down, and I'm going to see, you know, if I can shake this, you know, analyte off the sensor. I said, Hussein, that sounds crazy, but you can shake analytes off. Maybe we'll get Taylor Swift to write a song about it, and we'll have a new theme song. Turned out she already had written a song, but that's okay. So anyway, he did this, and lo and behold, so he applies 100 hertz, 300 millivolts, so not super high frequencies or anything. You can't go too high or it degrades the sensor. But here he is at his plateau, 1 minute of reset, he's back down to an empty sensor. So that's the solution. You reset the sensor back down to baseline, you let it re-equilibrate, and if the concentration has gone down, you can see that because you plateau at a lower level. So really, I mean, it's incredibly clever what he did in retrospect, the way these things always are. It seems like, well, of course that would work, you know, but this was a really, I think, a really insightful thing for him to propose. And it's super systematic. So these charts, this is like a typical Hossein experiment. He never goes into the lab and does one experiment. He always does like 1,000 at a time. And he stepped through frequency space and voltage space, or applied potential space. Blue means the sensor didn't reset. White means it's starting to reset. Red means it's totally reset. Okay, so you can see, so the top one is a low affinity sensor. So you start to walk across the voltage range and then you start to reset around here. And then you kind of go up in frequency and then you don't have to be as high of a potential. If you go down to a moderate affinity sensor, you can see that that zone of regeneration kind of moves because you have to put more force into the system to get the sensor to reset. And then if you have a high affinity complex, you really have to kind of hammer on it. You got to be up at 100 hertz. You got to be at the higher potentials. And so if you think about what we're doing here, we're really just modulating KD by stressing the system, right? When we measure KDs, the molecules are just gently drifting through solution, right? Like we're not putting any force on them. They're moving, but not fast. Here, now we're doing this. And so the thermodynamics of that complex are different, right? There's other stuff going on that are affecting the affinity. And I've said we were totally switching gears, but here's one nice simulation, I guess. We did MD on this, and obviously when you're doing molecular dynamics, you're on a different time scale. There's a lot of things that are different than the actual experiment. But when we did MD and we introduced the oscillations, we could see the hydrogen bonds in the complex breaking. And so, right, we're dragging the complex through solution. I don't know if they're breaking because of the force and the drag or the water molecules that they're colliding with at a higher frequency. It could be either of those things. But we're breaking hydrogen bonds, so we're subtracting a few kcals out of the complex. And so that's how the KD is changing. It's because those interactions are being disrupted.
This is with an aptamer?
This is with an aptamer, yes.
Does it work for an antibody?
Yes, it does. It does.
Have you tried a nanobody?
A nanobody? We've made sensors out of nanobodies, but I don't think we've reset them. It works on the EAB sensor, the Plaxco sensors that I showed you earlier. It works on those. We had an insulin antibody that we tried. It worked on that. So we have maybe 4 or 5 examples in the paper, but not an antibody. But we should try it because we have one in the lab. Okay, so then, so this is how the measurement goes, right? We're just kind of measuring, we reset. Here we're pulsing in different concentrations, and so you can see that we're able to do that out after we reset, so we can go up, and then, just keep resetting and then go down, and just kind of up and down, up and down. The sensors don't degrade as long as you stay in this potential and frequency range. You don't see a lot of signal loss, which is obviously very helpful for continuous monitoring. But we can kind of go and go and go for hours or days even. And oh yeah, here's the data. These are both aptamer sensors for IL-6, TNF alpha. The insulin one is an antibody. So it seems to work the same way. The magic voltage and frequency is different for every sensor, right? Because it depends on the KD. Okay, so then let's get these things into the ISF. Let's do some real continuous monitoring. So Hossein made these little microneedles that are basically just 3D printed housings, and then he's got 3 electrodes that he kind of puts into this. It's got a ledge, so you can just put it right through the skin, but you don't go into the blood, you stay in the interstitial fluid. So he had the right device to do this. I already told you why inflammation is important to monitor, so he decided that was going to be the application that he went after. And so what he ended up doing was measurements where he went into a diabetic rat model, very well accepted in the diabetes field, monitoring IL-6 and TNF alpha over time. He also took blood samples and took ISF samples out of the rats so that he could benchmark against those measurements. And this is kind of all the data. I have to break this down so it's a little bit easier to look at, but basically you've got IL-6, TNF alpha, fasting plus an insulin injection, and then fasting and an injection of LPS to induce inflammation. These are our measurements. These are the measurements in the ISF that we took out of the animals, okay? And then we analyzed it by ELISA. So here the animals are fasting and their inflammatory cytokines are drifting down. That's what you would expect. Here, what's really interesting, I mean, the ELISA data is not all that interesting. It's just, we're just kind of taking time points and the trends are more or less the same. But here, when we inject insulin, you can see these little blips in the traces. That's because we just poked the animal with a needle, right? And you get a little bit of inflammation from that. We can see that. You can't see it with the ELISA. The time points are too far apart, so you just can't sample fast enough. And then when we do the LPS injection, then the inflammation really starts to take off in these animals, and we can see that. So this was a really nice demonstration. I mean, it was a huge amount of work that Hossein put into this to, really get these measurements to be informative and reproducible, but it now gives us just a great starting place for studying the effects of all kinds of things, diet, exercise, stress, you name it, and seeing how that impacts inflammation in living organisms. And this is not important. This is what, if you're an analytical chemist, to really convince people, you have to do something called a Bland-Altman analysis, where you take the gold standard data, you take your data, you do a statistical comparison, and you have to kind of come out of this with the two methods being comparable. We did that. Thank you, reviewer number 2 for asking us to do that. But it was nice to see things pan out. And so where are we going? We have this technology embedded into several large-scale projects funded by DARPA or ARPA-H, where these are devices that are kind of looking at things like circadian rhythm and trying to stabilize circadian rhythm for military folks. This one is an implant that maybe someday will go near a tumor to kind of sense the microenvironment of the tumor, but also treat. We have several projects on chronic disease monitoring, and then maybe most importantly at the Biohub, because that was really the inspiration for all of this. We are making these tissue sensor hybrids where we're embedding really fine electrode meshes into human tissue models and then measuring cytokine profiles over space and time. So that was really the driving force. I mean, there's the continuous monitoring application, but we also really want to make these hybrid systems with the Biohub, and we need the sensing technology to do that. Okay, so I just want to wrap up and again thank Hossein and Javadomoy and Sarath for all of their incredible efforts, as well as everybody else in the lab. And thanks so much for having me.
Questions for Shana?
Thanks for the great talk. It was really nice to see the whole evolution, kind of when things don't go well and when they do go well. So thank you for taking us on that journey for sure. Just a question about the concept. Your postdoc, and I'm blanking on his first name right now.
Hossein.
Develop. Is there, I guess, have you also looked at joule heating as a function of that, or could you use temperature modulation to change, I think, maybe the entropy of that interaction and change the KD?
Yeah, potentially. We've thought about that over the years. It's a little hard to get the precision that you need, right? To do like one and not the other. We've got that DNA arm there. It's closer to the electrode, so we don't want that to get, we don't want to mess with that. We also had a lot of interesting discussions with our reviewers about why they thought the sensor was resetting versus why we thought it was resetting. And there were quite a few experiments that were done to really kind of like reinforce our mechanism. It's just a mechanism, right? And mechanisms just exist to be disproven, right? So I think we need to really dig into it a bit more. One very interesting result we got on the mechanism was that we did some experiments in higher viscosity, like in glycerol. And we thought, okay, that should modulate the reset parameters this way. It went the opposite way. So there are some effects that we still don't understand. We think there are some inertial forces that are probably in play. And there's, these kinds of systems every experiment you do, right? The mechanism may shift a little bit. So I don't think it's heating, but I would not rule anything out.
Thank you. And I guess, as an engineer, I was kind of taking it from another perspective which was, if Satan has a perturbation that he applied, what other perturbations could you? So that's kind of a neat aspect.
Yeah.
Thank you.
It is neat. And there's another student in the lab who actually, instead of shaking, he like mushes the complex into the electrode, and that also gets the sensor to let go of the protein. So yeah, there's a lot of ways to disrupt these things.
Thanks for the beautiful talk, Shana. Like you said, it's a beautiful combination of the chemistry and engineering attitudes. Mine comes back to the shaking, and it's related to both Paul and Ing's questions. So you said you were thinking about the inertial forces. So you said it's dependent on KD, but then did you also look at not only KD, but the size of the load that you're attaching? Is there a dependence on that of how hard it is to get the load up? Because my quick guess was a smaller load is harder to shake off than a large one.
Yeah. Absolutely. And we have thought about, I'm just thinking back to some conversations, we've definitely debated it a bit, but I don't think we systematically, because you have to get everything kind of the same, right? So I think to do that right, we need like one receptor that binds a protein that comes in a few different splice variants or something, and then we can do that. That would be really interesting. And I also want to get at this. Is it drag force? Is it collisions with water? That I think will be interesting to chase down.
Yeah, because my naive 5-second hypothesis was just that, it's just competing competition of the drag force with how much hydrogen bonding.
Yeah.
So
yeah,
it seems.
Really
Thank you again.
Anybody else?
What is like the biggest factor that goes into like the lifetime of the device? Like over time, is there like fibrosis around the electrode, or is there just degradation of the electrode?
Great question. Yeah, this is the hard part that nobody writes science papers about, right? How do you actually get these things to be stable long enough to realize all the applications that I was talking about? And we don't know yet. The animal experiments that I showed you, and I should have mentioned this, these animals are under anesthesia. They're lying still because it's a huge problem to let them run around, right? They're wired to the other stuff we need to do the experiment. So we couldn't leave the sensors in long enough to see if there was degradation, just because you could only leave an animal under anesthesia for so long. But we know, fibrosis is absolutely a thing. All implanted devices are going to have some level of fibrosis. So, the next phase in all of this is what coatings do you need, right? To kind of cut down on that. How do you need to protect all these biomolecular components so that they don't get chewed up by nucleases and proteases and all of these, enzymes? So, in some of the projects like the DARPA and the ARPA-H project, like we are being steered into doing that. But it is a solved problem. Like the continuous glucose monitor, like it works, right? And there was a ton of work that went into stabilizing it, right? So I don't happen to know what they did to stabilize it, but I would love to not reinvent the wheel and, use whatever solution, at least as a starting place. So we'll see, but you're absolutely right. That's a whole other set of issues that we did not address here.
Thank you.
Yeah, hi, thanks. It's a great talk. About this, this shaking mechanism where you're dissociating things. you topped out at 100 hertz. Is there any reason for that? Or it's just as high as you wanted to go, or naively I would expect you to go higher. And it seemed like it's more effective.
Yeah, you're absolutely right, but what we observed, if we went higher than 100, the sensor started to degrade. So the signal started to disappear. I don't really understand why. I don't, there's no chemical, physical reason that I can come up with. The voltage range for sure, like you can't go outside of a certain voltage range because the thiol gold bond that we use to anchor our sensor starts to degrade. So that makes sense, but I don't understand the frequency limitation. We are going to start making new constructs that, let's say, have multiple thiols. Maybe that will help them be more mechanically stable. And then we could go to higher frequencies. Maybe then we could shorten the amount of time that we need for reset. So it's definitely something worth looking at.
Thank you.